We're pleased to have you join us for the Southeast Ocean and Coastal Acidification Network State of the Science webinar series. The title of today's presentation is Deciphering the Effects of Ocean Acidification on Microbial Assemblage Structure and Community Function. This webinar series is hosted by the Southeast Ocean and Coastal Acidification Network, or SOCAN. These webinars lay a foundation for conversations about state-of-the-science ocean acidification science in the Southeast region. The webinars are intended to create a dialogue among scientists to identify what is known, what isn't, and what research in other regions of the United States can be applied to better understand ocean acidification and its impacts in the Southeast region. With awareness of and access to research and its implications, webinar participants will be able to collaborate to better understand and adapt to ocean acidification moving forward. I'm Paula Keener with NOAA's Office of Ocean Exploration and Research and a member of the SOCAN Steering Committee, and I'll be moderating today's session. Jennifer Bennett-Mintz is Education Outreach Coordinator for NOAA's Ocean Acidification Program, and Jen is also on the line and will be helping to facilitate today's session. During the presentation, all audience members will be in listen mode only. You are welcome to type questions related to technical issues or questions for the presenter into the questions in the bottom of the go-to control panel on the right hand of your screen. We'll be monitoring these incoming questions and will respond to them or pose them to our speaker. We are also recording this session, and the video recording and the PDF presentation will be available after the presentation on the SOCAN website. We are very excited today to have Dr. Astrid Schnetzer with us from North Carolina State University Department of Marine, Earth, and Atmospheric Sciences. Astrid received her PhD in marine biology from the University of Vienna in Austria and conducted her research at the Bermuda Institute of Ocean Sciences. Astrid is a biological oceanographer interested in plankton diversity and ecology, food web dynamics, and harmful algal blooms. Astrid examines how environmental perturbation can impact diversity within plankton assemblages, and by that, she looks at plankton community structure. So Astrid, I'm going to hand control of the presentation over to you, and you are welcome to begin.
All right, I hope you can hear me. Thank you so much, Paula, for the introduction. And thank you so much for giving me an opportunity to contribute to the SOCAN webinar series and just plain to talk about some of the things that I love to research and that fascinate me. And my research, as you stated, is in regard to plankton communities and the processes that drive their abundance and community structure. What you see on the first slide is a couple of names, collaborators that I've mainly worked with on the West Coast a couple of years ago, and I was fortunate to have my first experiences in ocean acidification research with this group of researchers that included Dr. Karen Hutchins, Dr. Fu, and some extraordinary students and also postdocs like Avery Tedders. I'm going to jump in sort of the outline into stating my ocean acidification wish list, and I shouldn't say it's mine. It's one that many people have on their mind, especially people who study biological systems and communities, and then use that wish list to make a couple of comments in regard to what it entails to study microbes. This is something that is probably very obvious to those of us who look at small critters in the ocean, but not necessarily for people that operate or study on a larger scale or with larger organisms. And when I say microbes, the organisms that I am studying are mainly protists and microzooplankton. So you see on the lower panel a couple of these organisms. We have some major groups of phytoplankton like diatoms and dinoflagellates, and then some additional pictures of microzooplankton. So phytoplankton being the main primary consumers, as we're aware, and protozoa being the main primary consumers, as in coastal areas they are able to devour up to 75% of that primary productivity. So we also want to know how they're affected by these changes in our oceans. Then I would like to give some examples where we're beginning to decipher connections, and those are from some lab studies with single species, also natural assemblages, and then I would like to talk very briefly about some of the mesocosm studies and what they're able to show us. And where from here will lead me to a couple of final words in regard to what I think that will be very promising for the Southeast region, and also a little bit more of my very private wish list in regard to where we might want to take this. So the OA wish list, again, one that has been mentioned and reiterated by many in the field, goes in regard to some of the knowledge gaps that we're starting to fill up, or fill in, I should say. There's a strong push towards going and studying single species, but now also mixed communities, since how they behave under environment perturbation or increasing CO2 isolated in a lab is very different from what happens if you have species interact. And I will give an example of that from some of the study that we did previously. Another one on that list, I think, is certainly going from single trophic levels to functional groups like primary producers up the food web. And some of the mesocosm studies that have been done over the last couple of years have shown us tremendous insight in what it means to have anything involved from viral, bacterial, protist to zooplankton organisms within these experiments. Another one where we've made tremendous stride, and it's almost impossible to keep up with the list, so I only have a couple of these studies cited, and take a disclaimer that it's really hard to keep up with the number of studies that is coming out, which is exciting. Deal with multifactorial influences where temperature, light, nutrients, some of the more prominent ones are tested together with CO2. Another one that I am very excited about is that we're starting to get away from just looking at community structure, but also community functions. So in addition to looking at standing stocks or species diversity or community structure, we're now also looking at the fluxes and the processes. And for microbial organisms, we're making really large stride here with genomic approaches that are being increasingly incorporated, and it's very exciting. And then one that baffles everybody a little bit is how we can use these incubations that are typically done in short term meaning anything from a couple of days to a couple of weeks, to what will happen to these communities over long term. And a lot of the talk here is about acclimation. What are some of these organisms capable of dealing with naturally? What's the genetic pool there? What's the genetic variance? Versus when do they start to have some change in the genes and start to adapt to these conditions over gradual change. So just to demonstrate where some of the difficulty comes in, or the complexity with persistent organisms, is if I highlight these two wish list items from single to mixed communities and from community structure to community function. So when we study persistent assemblages, we typically look at organisms that are tremendously diverse, morphologically, but also in the range of sizes that we're looking at. So you have your little heterotrophic nanoflagellates, you have your diatom chain, and then you have something as huge as a forum or other sarcodines. So we're talking about organisms that encompass up to 4 orders of magnitude. Also, we're talking about organisms that change very quickly. We have some of these larger sarcodines where we have a few per liter, but as we know, with the example of a harmful algal bloom event, they can increase their abundances within days, and then you're talking about millions per liter. So the scope of which you have to cover sometimes within the system that you study is pretty Another one is that while they might be the same sizes and are the same taxonomic group, they don't all do the same thing. They have a different capacity of making a living and have several functional modes that they might use and rely on depending on what the conditions are. And I just want to put in a quick couple of words in regard to genetic approaches, which are now allowing us to get over some of these hurdles that I just mentioned. So if you think about these different taxonomic groups, if you rely on morphology to determine who's there, At what given time, you might be forced to use different preservation methods. You might be forced to use different techniques, from flow cytometry anywhere there to larger microscopy tools. And it might also be that some of these organisms don't preserve very well. Now, with genetic approaches, we're getting a little bit more independent, and we can assess entire communities and process higher number of samples to get at spatiotemporal scales, resolve those better. And we're also beginning to account for some members that are not necessarily abundant. So if you look at this graph right here, what this shows you, it's a rank abundance curve. You have the relative abundance of the microbes on this side, and you have your species list or taxa list on the x-axis. And what you typically find when you grab a water at any given time from a specific place is that there is a couple within this part of this rank abundance curve that are very abundant. They're very obvious. And if you have a harmful algal bloom going on, you typically only find a couple that dominate this community. But what we've learned is, and this is also coming a lot out of bacterial research, and the term of the rare biosphere has been found from Sogan's group for bacterial communities that were studied, is that there's this long tail of rare species. So these are species that are there once or twice, they show up, but they don't seem to be very dominant. Now, depending on the method that you use, looking at these microbial assemblages, if you do microscopy, you get at some here and there. If you do culturing, you might get at some here and there, but it's always important to remember that we only have a small fraction of these organisms in culture because they don't tend to grow very well. And if they do grow in culture, they don't necessarily are the ones that play really important ecological roles in the environment. Now, as we use improving technology in regard to genomics, we can get out on that rare tail, next generation sequencing being the one that is most powerful. And with those methods, we can start to figure out what some of those roles are. But what we've also discovered is that there's new lineages and taxa out there that we haven't even accounted for yet, that some of those spatiotemporal dynamics are really quickly changing. And we have not yet penetrated the rare biosphere even with next generation sequencing. Now, does it matter who is here and what they might be capable of doing? That depends on your point of view. Some of these species, if you think about harmful algal blooms in coastal waters, you might have a sample where you don't even see a specific species that might become toxic or abundant because they hang out in the rare biosphere, but then this window of opportunity opens up and these take over the entire community. So it's important to know when they are there and when those community shifts happen. So genomics allow you to do that, but not only that, they also allow you to link to functional parameters, which is very exciting. And as we get more and more genomes sequenced, we can use those to see what's really going on in the environment. So I mentioned that one of the tricky things in regard to microbial assemblages can be that while they might fit into the same taxonomic group or the same size range, they don't necessarily do the same thing in regard to trophic. So what you see here is, again, 4 orders of magnitude for these different protists. Also included here is the bacteria and the cyanobacteria. And what I want to point out on the x-axis is that all of these guys, all of these critters in these different size ranges are phototrophs. On this side, we go over to the heterotroph, things like ciliates or nanoflagellates. And then in the middle, we have this increasing number of species that we now know that are actually mixotrophs, and they can switch between being photosynthetic or versus getting their nutrition by feeding on other organisms. And this gives them a broad ability to react to changes in the environment or food environment. And this is all things that we have to sort of consider if we look at how community function within these microbial assemblages might change with CO2 increase. Just to alliterate, so here the arrows here that indicate where the carbon is flowing or who's eating who. The black arrows show consumption of organisms by mixotrophs, and the green arrows show the consumption of organisms by heterotrophs. So it's kind of confusing, a little bit complex, but it also includes the red arrows, which show smaller organisms preying on larger ones. So it goes up and down in regard to size ranges. So if we think about a rank abundance curve again, you have your community here. You know who are the abundant ones, the most abundant ones under certain circumstances. You have your rare species. What you have happened is you have a change in the environment, again the example of a harmful algal bloom, and then you have reshifting. Some of these organisms might benefit from this difference, some of them might just decrease, and you can play this over and over again. And this can happen within hours or days of a community being tweaked or just simply a microbial assemblage being put into a bottle without any further parameters being changed. So what you end up with in this example is you have some driving forces. This could be CO2 temperature or other changes, and you end up with these communities that are very different in community structure. Now, one of the main questions that we're trying to grapple with is, does that mean that ecosystem function or ecological processes as a whole are actually changing? Or do these, let's say this is a phytoplankton community, do these primary producers come up with the exact same primary production rates as these do? If that's the case, it might just really not matter, right? So if we have these different forcings like CO2, layover temperature, light, salinity, nutrients, where do they pull the community, and what does that mean for the overall community function? And you can think about this as being a primary productivity rate, a chlorophyll rate, or whatever else you want to think of. So now I want to move on and show you some examples of what we did with mixed assemblages early on in the Southern California coast. And what this is a layout of is some of the experimental setup that we did. What you see here is the pie chart that shows you the contributions, relative contributions of these four major dinoflagellates. We collected a mixed dinoflagellate community that was strongly dominated by a red tide forming organism, Lingolodinium, and we incubated them in semi-continuous batch cultures for 2 weeks at low CO2, ambient CO2, and high CO2. And then after 2 weeks, we just sampled and looked who of these organisms were the most dominant and if there were shifts in regard to their abundance. And as you see in this pie chart is that a general trend with low CO2 to high CO2 was that Lingolodinium, which dominated very strongly at the beginning, actually lost some of that share. And some of these other organisms, which also included a toxin-producing Alexandrium, actually gained some of their community community share. So after that 2-week incubation experiment, the next step that our group did was, and this is a picture of Lingolodinium and the red tide, you can see how they color the water when they're dominant. What we did next is we went and isolated these organisms from the different final incubations and grew them under the conditions that they were most dominant or thriving the most as single cultures and allowed them to grow under those conditions for up to a year. So we took from the mixed communities what we considered the winners and put them isolated under those conditions and let them go at it for 30 to 120 generations and allowed them to adapt over longer time scales. After we did that, so this is the time frame right here, this is the initial experiment, we competed them. We put all four, after they had a chance to be conditions under those elevated CO2 regimes, after 4 months, 8 months, and 12 months, and just repeated our competition experiment right here. And what you can see, there's a couple of things to take away from this is that some of the members within this community did well no matter what. So Prorezentrum P. micans right here, the red pie chart pieces had a huge gain throughout the time that they were allowed to be within culture. Now, Prorezentrum could be considered something like a lab weed that does well anyway, so this might not be tied to any of the CO2. What you can also see is that some of the organisms like Lingolodinium seem to consistently do better under low conditions of CO2. So you see this trend not only in the initial incubation, but also after 8 months, where the blue part of this pie chart decreases with higher CO2. So some trends were repeated after a while, and some trends showed also that it might tremendously depend on who you're starting out with, like Perizanthrum in this case. The shift towards potentially toxic organisms, which is both Alexandrium and Gonialux, was interesting too. So if you could consider Lingulodynium a harmless or non-toxin producing, harmful, non-producing algae, now we're talking about a community that is to a larger degree made up of those potentially toxic or harmful species. This shows you just Bray-Curtis similarity for each of those. Again, here we have the triplicate incubations under low, under medium, and high PCO2 conditions after the first 2 weeks, then when we recompeted after 4 months, 8 months, and 12 months. And although they were all significantly different due to CO2, what we found is that over time, with increased acclimation or conditioning period, the differences of CO2 started to weaken. So the longer they were allowed to adapt to these conditions, the less the CO2 seemed to drive what happened within those two weeks of incubation. The other thing is that points towards, and others people have emphasized this over and over again, is that it's really important to conduct these types of competition experiments with organisms that are freshly isolated and not just kept in culture over long periods of time. So you're also looking at clonal isolates that might not be very representative of the genetic variants that you have within the field. So with genomics, which we did not do in this experiment, you will be able to actually look at the different strains and better get a feel for whether you have genetic variants within your isolates. Another thing that we looked at in this experiment was that we measured growth rates. So now we're coming from community structure to looking at, well, how well did they do under the different CO2 conditions. And what you can see here is we did growth rates in the mixed communities in the original 2-week incubation, and then we did it for the mixed communities after a year. And this one in between is for the single isolates by themselves without being put together. And one of the striking things that we found is that there's way higher species-specific growth rates for these when they are put together. And thinking about it, it's not very surprising, but very important that there's interspecies-specific interactions that might contribute to this huge difference between a mixed growth rate versus growth rates within a single culture. As I mentioned before, you have allelopathy potentially going on. There was some toxin production from Alexandrium, allelopathy meaning that it can release dissolved toxins and inhibit the growth of its competitors. We have mixotrophy. All 4 dinoflagellates were potentially mixotrophs and could have fed on each other, which would have gained them a tremendous nutritional advantage. So these sorts of interspecies species interactions come heavily into play if we look at these sorts of cause and effect relationships and mixed communities versus single cultures. This was one that we did for looking at mixed communities under different CO2 conditions. There's a growing number, like I said in the beginning, of multifactorial studies where you look at CO2 and temperature, where you look at CO2 and nutrient availability, CO2 light and iron availability, and what all of these have started to show is that there are species-specific responses that could have true implications for the entire food web as a whole. Marked in red, there is a very nice review from Fu and Hutchins' group where they show that several of these isolates for toxic algae, when they grow them under increased CO2, combined with some sort of nutrient limitation, that the toxicity actually increases for these specific strains. So not only might we have an increase in how much harmful algae contribute to communities, but also how toxic they might become. And some of the interesting studies that looked at different diatom communities have shown that there might be a shift between more or less silicified diatoms, which would have tremendous impact on export flux of organisms, whether they can sink or not sink from the mixed layer. What's really exciting, this is a very new study from Stephanie Tukkiewicz. She presented this at the OEA PI workshop not too long ago, where they took some of these lab results from single species, but also long-term experiments and mixed field incubations. And what they were looking for was conditions where you had ambient and increased PCO2, and they came up with the growth rate response under these different conditions. And on this side of the curve, this is the growth rate response. Above 1, you can see that they reacted positively. Below, they had a negative effect with increased CO2. And you can see you have different groups here: coccolithophorids, diatoms, other large phytoplankton, diacetrophs, Synococcus, and Prochlorococcus, so an array of cyanobacteria included in the study. And here's the range of growth responses you can find within each of these taxonomic groups. Some of these growth responses might have landed on the upper or lower side, even for the same species, but specific strains. So there can be a lot of genetic variance going on in here. Now, they took these data, I think it was a total of over 150 observations, and for these varying functional groups, and they looked at 16 or grouped them into 16 functional groups that also had differing temperature ranges and growth optima based on nutrient and light dependence. And they embedded them into an Earth system model and ran how they would probably do with increased CO2 from using a business as usual scenario from the IPCC. And then they were focusing on community structure response and also some of the biogeography, how that would change. And I'm trying to stand very close to my notes here because my disclaimer is I'm not a modeler, so I'm trying to be very careful. So what you see here is the same sort of summary where you see these different functional groups. We got coccolithophorids, diatoms, and different cyanobacteria groups in regard to their response again and the growth rate response. And the colors up here in this figure correspond to the colors that are maintained in these graphs down here. What you see down here is the models being run under different scenarios. So what you can see on the very left here is the model run with all of the different drivers, which includes physical change or physical drivers as well as CO2. This one includes only physical changes and CO2 is kept constant, and this one turns it around with showing PCO2 being changed according to those 2100 scenarios and the physics not changing. And when I say physics, what they used was temperature, circulation, and sea ice. What you see here is the predicted change in biomass. The symbols within each color shows different species within each of those functional groups, and they used 4 of them incorporated into this model. And you can see that overall, right here, the biomass fraction less than 1, it indicated that there was a biomass decrease, while at a biomass fraction larger at 1, you would see a biomass increase. Now, if you compare the scenario run with all drivers, physical and CO2, to that only includes the physical change and that only includes the CO2, is that they found that for some of these, the main driver might actually be CO2 rather than any of these other parameters. Now, many of these data points obviously could be flushed out with additional experiments where you look further into interspecies competition, either within the functional group or between those functional groups, and also over a larger range of probable CO2 concentrations. Again, these were taken from experiments that just had 2 different CO2 treatments. And the other thing that we obviously have to do a better job in feeding into these models is getting information on some of the synergistic and antagonistic relationships between those different stressors. But I think it's a fantastic example of how we get close in using these varying experiments and putting it into a bigger picture to start to understand how this might affect different functional groups and different primary producers. If you take it a step further, this review is a little bit older, from 2009, but it shows really nicely how we are concerned in regard to not only distribution and abundance change, but also the quality or the characteristics, the elemental composition of these phytoplankton. So again, in the upper graph, you see a compilation that shows you the percentage change in CN and CP ratios. And what you can see from the different experiments that were conducted with single species is that generally they tended to show an increase in CNP, or they were not affected. Now, this is single cultures. If you go now out and you test this with natural communities, and you can see this is a whole array of different environments right here that are listed in this compilation, you see that the picture becomes way more complex. Obviously, if you don't look at a single organism but mix them together, all of these community changes and interspecific relationships come to bear. And I certainly think that there is a tremendous need for looking at these community-based ones together with the specific stoichiometric impacts depending on where you are on local or regime-specific scales. Why does this matter? Well, if you think about the trophic food webs and how this, so we're trying to go from, throughout this talk, from going from lab studies, the single species, to mixed communities, and now go up the trophic levels. This is a way to, this is a really nice review by Hutchins and Karen that was focusing on how microzooplankton might be affected by CO2 increase in aquatic systems. And just to remind you, the microzooplankton, the ciliates, the heterotrophic dinoflagellates, and so forth, might be consuming up to 75% of the primary production in coastal areas, so they're really truly way in. And we do not really have a very good grasp on how they're affected. So I'm inviting you to take now a microzooplankton-centric view, or grazer view, and if you think about the microzooplankton, there's certainly effects that might be direct for them that have to do with intracellular pH, enzyme activities, and so forth. And then you have this whole array of bottom-up effects that might implicate how well they're doing. And these come from some of the changes and shifts that we talked about up to now in the phytoplankton. And this was changes in primary productivity, altered stoichiometry, or in this case, prey quality for the microzooplankton, and also taxonomic composition if we think about potentially harmful algae. And so bottom-up plays a role, but also top-down. We also now starting to get a better feel of how larger grazes might be impacted in community structure and how trophic connections between these different levels might shift. That's very complex, and one of the things that is best, I think, been done so far to look into this is some of those mesocosm studies that have been conducted. And many of you might be familiar with some of these. This is taken from Ulf Rubesel's summary of one of their pelagic ecosystem CO2 enrichment studies. And what they used in these studies are these large mesocosms that penetrate, I believe, down to 20 meters in the water column or so. And they enclosed them, changed the CO2 levels throughout a large gradient. You see right here, the bluer colors are the lower CO2 treatments, intermediate is gray, and then red is the higher ones, and they range from 175 to over 1,000 ppm. And what they do with these mesocosms is that they truly enclose an entire planktonic community, which really allows to look at some of those larger scale effects. What this one shows you is chlorophyllate concentration right here in each of those mesocosms. Again, the red colors are the highest CO2 treatments, intermediate in gray, and blue is the lower CO2 treatments. And then they have these varying phases, and the phases refer to nutrient additions that were made after 4 days in the mesocosms and then after 13 days. And you can see the responses in chlorophyll falling right after. So they look at the system as a whole. They look at primary producers, and standing stocks here are chlorophyll A, pico, nano, dinoflagellates, and diatoms. And then they also look at the grazers, micro and mesozooplankton. And then they also look at the rates. We have right here net carbon uptake, primary productivity, and then some other measures. I think they were done with oxygen change, oxygen concentration. And then we have zooplankton grazing, and also information about carbon experts. So very complex, very large, multi-investigator experiments, but really a fantastic way of looking at how the system might be affected as a whole. And a couple of things that I want to point out here is that if you look at the chlorophyll A stock, what they show here in these different colors for phase 1 through 3 is whether they were positively affected by elevated CO2, negatively, or there was no measurable effect. So one of the things that you can deduct from this is that some of the phytoplankton did better than others. So you have an increase in phytoplankton towards phase 2, especially in the smaller pico and nanoflagellates and also the dinoflagellates, but not necessarily the diatoms. And as phase 3 comes around, you actually have the silicified diatoms not being favored at all, which will also feed into the carbon expert flux that is shown as being negative towards phase 3. Again, diatoms having silica frustals or shells are the ones that typically tend to sink out as the bloom dissipates. And you can see that the main winners here are nanoflagellates and dinoflagellates towards the end of this experiment. Also interesting is that there was no measurable effect in microzooplankton and mesozooplankton. And there's one of the papers that is associated, there's many papers associated, this is just a review summary that gives a broader picture of these results and trends. One of the studies that just looked at microzooplankton was published by Archer and his colleagues, and it showed that there was no change in phenology. They showed that microzooplankton were generally reacting by increasing if there were more phytoplankton, but there was no species composition shifts, at least not within those 30 days or so of the mesocosm, which might point just to the fact that microzooplankton grazes might be way more resilient to deal with changes that are induced by increased CO2. The summary also showed a really nice diagram that sort of, again, shows some of these major trends and points out how systems can be affected overall. So if you think about this being the status quo, present CO2, it's a fairly oligotrophic system. Again, it depends heavily on where you conduct those, and the addition of nutrients might just be a tremendous shift for these systems. You can see that at present CO2 or ambient CO2 in this example, you have your incorporation of carbon into phytoplankton biomass. You have some dissolved organic matter being produced, DOM, you've got some pyrolysis going on. Some of the dissolved organic material is shuffled back through the microbial loop to the higher grazers, a little bit of sinking going on. Now, if you change this situation simply by adding some nutrients, you can see that the flux is mainly changed in regard to some more sinking towards out of the mixed layer. However, if you now transfer that into an elevated CO2 environment, what they have found, or the trends that they've seen, is that there is a lot more of the primary producers leaking carbon into the environment, or some of the leakage going into the dissolved organic matter pool. But this is not being shuffled back through the microbial loop because they believe that the bacteria were nutrient limited. And this also had a negative feedback on the grazing. Now, if you put the nutrients back into the equation with the higher CO2, you can see that some of the microbial loop sets back in, steps back in, and some of the DOM actually gets incorporated better into the POM and the grazing, turns it around, and some of this carbon is actually shuffled or funneled off the trophic layers. But sinking of material is actually decreased compared to the situation where you only have nutrients amended. So it's pretty complex. Just look at these overall shifts due to these amendments, but there's no way, if you don't try to conduct these mesocosm studies, to really get a grapple of how this might propagate through planktonic communities. So where from here? I know this was maybe a lot of information for some. It's not enough in regard to how much has already been produced on information, and there's increasing review papers that help to really give a good idea of where we're at for different systems. But if I would look at that filling in the knowledge gaps, I think we're doing a good job overall. And my personal wish would be to really strongly push for the incorporation of genetic approaches to determine some of the structure and function changes, and also being able to look at single species and mixed assemblages, and also confirm some of the trends that we see for single species experiments. Does that hold true as soon as we go into the environment? The other thing that I think that is going to be extremely important and will help a ton is if we start to combine some of those lab and mesocosm experiments over the environmental gradients that we have within the area. And this is offshore to estuarine environments as well as along the coast from North Carolina to Florida. And if there's a lot of collaborative efforts that try to address these gradients, that we have a really good chance to standardize some of those response variables that are needed to put them into larger scale models, which I think is a great way to start to understand better what ocean acidification is going to do. And if I was very bold in regard to my wish list, we all know that SOCAN's mission statement includes that we want to set regional priorities for monitoring research. I would say we should consider something like the Southeast Acidification Laboratories, or SEALs, the name is debatable, that we have on our coast. There are similar facilities in Friday Harbor. They have them in Norway. There's scale-up versions of these somewhere else on the coast, but we do not have such a facility. And I think it could be tremendously helpful if we considered studying some of the very specific environments along the southeast coast that will allow us to go over different gradients of nutrient availability, light, salinity, looking at coastal environments as well as offshore, using these system approaches and mesocosms. And it would be tremendous help to get from our little culture experiments and microcosm experiments to larger scale understanding of OA and what it will do, and tie that in with all the field observations that are already being done along the coast. And it could create a hotspot for many of us to do system level research, and it could tremendously benefit from all the existing knowledge, like some of those SOCAN webinars demonstrated very well, that we know who the key organisms are, we know what the communities are that are important, and also what the pertinent scales are, whether you go inshore with high variability or offshore with lesser variability. It could be a great platform, and SOCAN can fully be leveraging that to talk to stakeholders and make them part of some of these research setups and designs. And I also think it would be tremendously helpful to have a place or a hub where we could have our students learn some of these proper techniques. I know there is workshops available. I know that many of us have gone to those to learn how to do seawater carbonate chemistry and try to do this OA research. But I think if we had it here, it would really help us to answer some of those questions. And last but not least, I would really like to have an ability to better answer my 7-year-old in regard to what OA all means. And I think we have a tremendous opportunity to do that if we had such a facility along the coast. And that's pretty much it. Thank you very much.
Astrid, thank you very much. That was fantastic, and I love the way that you presented your final concluding slides, seals. I think that's really something we should think about. So with that, what I would like to do is open the floor for questions to Astrid, and we already have one posted. And Matt Woodsock would like to know, how does increased CO2 that you mentioned as a factor in future phytoplankton populations affect geochemical cycles in our oceans, more specifically the nitrogen cycle and nitrogen fixation?
Can you still hear me?
Yes, Astrid, can you hear me?
Yes, I hear you now.
Okay.
I caught part of that question. It was about nitrogen cycle?
Yes, he's wanting to know how increased CO2 affects geochemical cycles, specifically the nitrogen cycle and nitrogen fixation.
There's a tremendous wealth of information coming from single culture studies on nitrogen fixation, and there's also a very new paper that comes out of Hutchins' group where they actually showed some of these. So I did not talk about, I have to admit, I did not talk about cyanobacteria in specific, but they showed that with some of these long-term acclimations with some of those cyanobacteria, that there might be irreversible effects on nitrogen fixation rates as these organisms are allowed to condition to them. I'm a little bit out of my comfort zone talking about the overall global implications for the nitrogen cycle. But since cyanobacteria are some of the organisms that might be most affected, there's a lot of information already out there.
Thank you. Astrid, can you comment on the use of continuous plankton recorders in regards to filling in some of the knowledge gaps on a larger scale, possibly placing them on ships of opportunity, etc.?
They're a tremendous help. I think that together with the genomic approaches and the large-scale sampling that has been conducted, especially over the last couple of years, I think the most challenging thing will be to try to find how they relate to each other and how you can join them or correlate them, since you have many of the phytoplankton still being mainly defined by their morphology, and we're only starting to put genetic signatures to most of those organisms that we already know.
Okay. Can you comment on the amount of labor involved in processing a single sample from a continuous plankton recorder, say per hour that it is in the water? Just to give an idea of, I have no clue what's involved in processing those samples.
I don't know either. I've worked with samples. I've not done it.
Okay, great. Okay, thank you. Thanks. Do we have any other questions for Astrid? Oh, here's one. I'm sorry. Can you share a bit more about your idea for the setup of, or excuse me, about what you know about the setup of your the seals concept in Norway?
Oh, I was just referring to the mesocosm studies that they have a mesocosm facility in Norway that is very similar to the one that is currently functioning at Friday Harbor on the West Coast. It's approximately the same scale, and a lot of new research is coming out of that. What I meant by saying that I think we should have one that is about approaching the same scale that they can study at in both of those environments. And the European capabilities in regard to having these floating mesocosms that are also used in connection or in collaboration with some of the US researchers is really something that I think we should try to live up to or try to get to. I think that we should truly consider a fleet of floating mesocosms and field station mesocosms, but that's probably a very big wish.
Mm-hmm. I am really intrigued by your idea for the seals, and I think that there is, well, there certainly is transatlantic implications for that as well, certainly through some agreements that are in place between the United States, Canada, and the European Union. So very exciting. So any other questions for Astrid at this point? Okay, well, Astrid, thank you very much for a fantastic presentation, and it just really demonstrates the complexity of what we are dealing with ocean acidification at a very fundamental level. So thank you so much. And I would like to, hello? Okay, and if we didn't get to, if you have questions that come to you later about today's presentation, please feel free to contact Astrid via the email, which you can find in the right-hand side of the screen of your control panel in the chat box. And if you're interested in joining the SOCAN listserv to learn about future updates and the Acidification Network, please sign up at the URL shown on this slide. And also, please direct any questions about the SOCAN network to socan@SECOORA.org. Thank you for attending the webinar today. We welcome any feedback, any further questions that you have, or suggestions for topics about this webinar series. You can submit input by replying to the follow-up email you'll receive or by simply emailing us. The recording of this webinar and a PDF of the presentation will be available on the SOCAN website. Our next State of the Science discussion will be on Tuesday, October 6th. We will not have one next week, so we will be skipping a week again. On Tuesday, October 6th, Zach Johnson will be joining us at 12 p.m. Eastern Time, and he will be speaking about temporal and spatial variability in ocean acidification from a time series off of the North Carolina coast. So we hope you can join us at that time. Again, thank you, Astrid, for your very informative presentation, and to you all for your thoughtful questions, and for also taking the time to join us today. We hope to see you at our next SOCAN State of the Science conversation. This ends the webinar.